Dear students, and today we are going to see the questions we can expect from the particular topic, the biomolecules. So as I told you, there are so many segments we have seen: polysaccharides, carbohydrates, nucleic acids, lipids and enzymes. And today we are going to see something about the questions we can expect from the topic, the nucleic acids and the related things. And see the first question. Uridine present only in RNA, that we know. Okay, uridine present only in RNA is a nucleoside, nucleotide, purine and pyrimidine. Okay, you have to choose the correct answer for the term uridine. We will recall the thing. What is nucleoside and nucleotide and purines and pyrimidine? As we all know, in case of nucleic acids, there are two types of nucleic acids we all know: DNA and RNA. Okay, so what is the major difference between these two things? DNA, that is refers to deoxyribonucleic acid, RNA refers to ribonucleic acid. Why we are calling this as deoxyribonucleic acid? Because over here the sugar, the ribose sugar is deoxy sugar, deoxyribose sugar. Over here the normal pentose ribose sugar. Okay, so this is the major difference. So apart from this difference, the one more major difference over here, as I told you, it is made up of the nitrogenous bases. There are adenine, guanine, thymine, cytosine and uracil. These are called nitrogenous bases. We can classify these nitrogenous bases into two major classes: purines and pyrimidines. Okay, so adenine and guanine are purines, thymine and cytosine and uracil are pyrimidines. Okay, so this is what you have to remember. So whenever you are talking about a nitrogenous base, you have to remember the term adenine, guanine, thymine, cytosine and uracil. But over here the term given to you is uridine, not uracil. Okay, so now we will come back to the other two terms, nucleoside and nucleotide. So what is nucleoside? So the nitrogenous base plus a sugar. As I told you, it may be a deoxyribose sugar in case of DNA, or it may be a ribose sugar in case of RNA. So the nitrogenous base plus the sugar is called nucleoside. Okay, so then what is nucleotide? Plus a phosphate group. Okay, by means of the phosphodiester bond. So as I told you, a nitrogenous base plus sugar plus a phosphate group. Together, these three together, they will form a structure called nucleotide. So the point you have to remember over here, whenever you are thinking about a term nucleotide, that is along with the particular phosphate group. If the phosphate group is present in the particular structure, that is nucleotide. If there is no phosphate group, only the sugar and the nitrogenous base, that is nucleoside. Okay, now you have to remember the terms. As I told you, whenever you are using the term adenine, that is refers to the nitrogenous base. Okay, you are not supposed to get confused with the terms. When you are using the term adenine, this refers to just the nitrogenous base. When you are talking about a particular nucleoside, adenosine, you have to use the term adenosine. Okay, so this is refers to nitrogenous base plus sugar, that is nucleoside. Okay, then what about the nucleotide? Over here you have to use the term adenosine triphosphate or adenylate acid. Okay, so these two terms refers to nucleotide. So the point you have to remember over here, there are different terms we are going to use, slightly different terms we are going to use for nitrogenous base, nucleoside and nucleotide. Okay, so the two things you have to remember over here, nitrogenous base refers to just the nitrogenous base, adenine, guanine, thymine, cytosine and uracil. Nucleoside refers to nitrogenous base along with the particular ribose sugar or the deoxyribose sugar. Over here you are going to use the term end with the particular INE. Okay, so adenosine, uridine, Okay, thymidine, cytidine refers to the particular nucleoside. Nucleotide refers to, over here you have to use adenylate acid or adenosine triphosphate or the uridine triphosphate, like that you are gonna use. So the point you have to remember, now you can choose the answer. Uridine present only in RNA is uridine is not a purine or pyrimidine. As we all know, purine and pyrimidine are just a nitrogenous bases. So you cannot choose this as a answer. Okay, it must be nucleoside or nucleotide. I told you, whenever you are thinking about a nucleotide, it should be come along with a particular phosphate group. So there are no phosphate group. This is not a uridine triphosphate, just a word uridine given to you. So this is not an answer. Uridine in RNA is a nucleoside is the answer. This is how you have to choose the answer for the particular type of question. And see the next question. DNA is composed of repeating units of, over here there are 4 choices given to you: ribonucleotides, deoxyribonucleotides, ribonucleoside and deoxyribonucleoside. Okay, so the differences over here: deoxyribonucleosides, okay, and deoxyribonucleotides, ribonucleoside and ribonucleotides. Okay, so as I explained in the last question, you have to remember DNA refers to deoxy. Okay, so DNA refers to deoxyribonucleic acid. Okay, deoxyribonucleic acid. Okay, so the point you have to remember over here, the major difference between these two, the presence or absence of the deoxy sugar. So the answer must be one among these two, deoxyribonucleotide or deoxyribonucleoside. We can easily eliminate the ribonucleotides. So ribonucleotide and ribonucleosides present only in RNA, not in the DNA. So the choices remain for you: deoxyribonucleotides and deoxyribonucleosides. So I told you, so DNA is a polymer. We all know DNA is a polymer. So polymer of nucleotides, so we can call this as polynucleotides. So what do you mean by polynucleotides? It is made up of the monomers of nucleotides. So there are so many nucleotides, it is being combined with each other by means of the phosphodiester bond, and they are going to form the particular structure called DNA. So the point you have to remember over here, the DNA is made up of deoxyribonucleotide. Okay, not side. Side always refers to without the phosphate bond or phosphate group. So whenever you are talking about a nucleic acid, whenever you are talking about a nucleotides, you have to choose the deoxyribonucleotides. So I'll explain you one thing. So we all know, we are calling this as ribonucleic acid and deoxyribonucleic acid. But I told you the major group present in a particular nucleic acid is a nitrogenous base. But why we are calling this as acid? Okay, so we are calling this as the major group over here is adenine, guanine, thymine, cytosine and uracil. Okay, this is the bulky group, the major group present in a particular DNA and RNA. But we are calling this DNA and RNA as a acid, deoxyribonucleic acid and ribonucleic acid. Why we are calling this as acid, not as a base? Okay, so the point you have to remember over here is the presence of the particular phosphate group. So in a DNA or RNA structure, okay, so the nitrogenous base is being attached with a sugar and sugar is being attached with a phosphate group. Okay, so the external surface is being covered with the phosphate group. The phosphate group can be ionized to form a acid structure, so that the nature of the particular DNA and RNA in a liquid environment, in a cytological environment, a body fluid environment, it is in the form of the acid. So that only we are calling this as deoxyribonucleic acid and ribonucleic acid. So you have to remember the thing, whenever you are seeing the term acid, you have to remember the presence of the phosphate group. Whenever you are thinking about a phosphate group, you have to remember the term nucleotide. Okay, so from this explanation you can easily select a particular answer. The question over here, the DNA. What is the DNA full form is deoxyribonucleic acid. So acid is present over here. So whenever you are thinking about acid, you have to remember the term, the presence of the particular phosphate group. So whenever the phosphate group is present, you have to recall the term nucleotide. So this is D. D refers to deoxy. So here you can easily select the answer deoxyribonucleotides for the particular question. And see the next question. ATP is a, okay, so choices are nucleoside, nucleotide, nucleic acid and vitamin. Okay, so among the choices we can easily eliminate the vitamin. ATP is not a vitamin. We all know ATP is a energy carrier molecule. It is going to provide the energy throughout the cell, and of course I told you ATP is a part of the particular nucleic acid. It is present in the DNA and RNA is in the form of, as I told you, A refers to adenine. Okay, so what is the full form of ATP? Adenosine triphosphate. Adenosine triphosphate. Okay, so it is present in both the DNA and RNA as a adenosine triphosphate. So the point you have to remember over here, ATP is a nucleoside, nucleotide, nucleic acid and vitamin. So I told you, whenever you are talking about a nucleic acid, it is talking about a polymeric structure of the particular nucleotides. Okay, so ATP is a single unit, so you cannot use the term nucleic acid for the particular single unit. Okay, so you have to choose the answer from these two choices, nucleotide and nucleoside. I told you the full form for the ATP is adenosine triphosphate. Whenever you are seeing the term phosphate, you have to recall phosphate is always associated with a particular nucleotide. Whenever you are seeing the term phosphate, then you can easily combine or you can easily connect the particular term with the nucleotide. So ATP is a nucleotide, not a nucleoside. Just the term adenosine is nucleoside. Adenosine triphosphate is a nucleotide. Okay. Now see the next question. Adenine is a purine, nucleoside, nucleotide and a pyrimidine. Okay. So this is a straightforward question but a bit confusing question. So adenine, we all know adenine is a kind of nitrogenous base present in a particular DNA and RNA. And you have to choose, there are no choice over here as nitrogenous base. You have to choose from the purine, nucleoside, nucleotide and pyrimidine. Okay, and we will start from this nucleoside and nucleotide. We all know it is a combination of nitrogenous base plus sugar plus, if the phosphate is present, it is nucleotide. If there is no phosphate, it is nucleoside. Okay, we all know there are two groups of nitrogenous bases, purines and pyrimidines. And what are the bases present in case of purines? Adenine and guanine. In case of pyrimidine, thymine, cytosine and uracil. Okay. So if the term is adenosine, you can choose the answer nucleoside. If the term is adenosine triphosphate or adenylate acid, you can choose the answer nucleotide. Now you can eliminate these two because the given term for you is adenine. Okay. So you have to choose whether it is a purine or pyrimidine. We all know adenine belongs to purine along with the guanine. Adenine and guanines are purines. Thymine, cytosine and uracil are pyrimidines. So the answer is purine. Okay. See the next question. RNA does not possess. Okay. So this is also a straightforward question for you. Uracil, thymine, adenine and cytosine. Okay. So I told you, again I will repeat to you, the major difference between the DNA, the presence or absence of the deoxyribosugar or the ribosugar. So apart from the first major difference, the second major difference over here, in case of DNA, the nitrogenous bases are adenine, guanine, thymine and cytosine. In case of RNA, adenine, guanine, cytosine and uracil. Okay, so you can easily see the differences over here. The purines are similar for both the DNA and RNA. So in both DNA and RNA, the purines are adenine and guanine. But in case of pyrimidines, over here in DNA, thymine and cytosine. In case of RNA, there are cytosine and uracil. So over here, the thymine is being replaced by the uracil. Okay. So uracil is only present in RNA. That is the point you have to remember. That is the thing you have to remember over here. So RNA does not possess, uracil is there, adenine is there, cytosine is there. Okay. So the answer is thymine. So thymine is absent from the particular RNA structure, ribonucleic acids. So this is how you can expect the question from the particular nucleic acid, and we are going to continue the patterns of question we can expect from the particular topic, the nucleic acids. See the next question. So in a double helix of DNA, the two DNA strands are, so over here they are talking about the double helical nature and the coiling nature of the particular DNA structure. Okay, so see the choices: coiled around a common axis, coiled around each other, coiled over a protein sheath, and coiled differently. Okay, so we all know DNA is made up of two strands. Okay, two strands of polynucleotides. What is polynucleotides? It is made up of the monomers of nucleotides. So what is polynucleotide? Made up of so many nucleotides as a chain. Okay, what is two strands? There are two strands of the particular polynucleotide. Okay, so see the choices over here. So in three choices you have the term coiled. Okay, you have to select the choice from here. Of course, all the four choices they are possess the term coiled. Okay, so over here, whenever you are talking about a DNA, the structure of DNA itself, you are not supposed to confuse with the protein. Okay, so the chromosome, chromatin, the nucleosome. Okay, so whenever using the term chromosome, chromatins, nucleosomes, you have to remember the DNA is coiled around the particular protein compound, histones. Okay, but when you are talking about the structure of DNA alone, you are not supposed to confuse with the histone. Here there are no proteins involved in the particular DNA structure. Okay, so the DNA structure is being proposed by the Watson and Crick. Okay, so according to this structure, there are no proteins involved for the particular DNA. Okay, so just you have to remember the DNA is made up of nucleotides. That particular nucleotides present as a polynucleotide strands. There are two polynucleotide strands. They are being coiled around each other. This is what you have to remember. So you can easily eliminate the fourth choice. Coiled around or coiled over the protein sheath is the wrong answer. Okay, so this is correct only in case of nucleosomes, chromatin and chromosomes. Over here we are talking about only the DNA structure. See the question again. In double helix of DNA, this is over here they are talking about only the DNA structure. Okay, so just eliminate the protein sheath covering. So see the remaining three choices: coiled differently, coiled around a common axis, coiled around each other. So all the three choices seems correct over here. Okay, coiled around each other and coiled around common axis, coiled differently are same. But over here they are not identical. They are similar terms, and you have to choose the correct among these three. So coiled around a common axis means there is a central common axis. Okay, around the common axis, that particular DNA strands are running around each other. This is what you have to remember. Okay, around the common axis they are running around each other. Coiled differently means that is some other term just opposite to the particular common axis. Coiled around each other is correct. When it is correct, when there is no coiled around the common axis, then you can choose the coiled around each other. But we have a choice over here, coiled around the common axis. Okay, then you can eliminate these two choices. So over here you have to remember in DNA there are two strands. Those two strands coiled around each other. Up to this is correct. Coiled around each other in a common access, then you have to choose the choice the A. Okay, and see the next question, which is not consistent with the double helical structure of DNA. Okay, so over here there are 4 choices given to you and you have to choose which one is inconsistent with the particular DNA helical nature or helical structure. So the choice 1, this is not a hydrogen bond we have mentioned over here, this is equal to, adenine is equal to thymine, cytosine is equal to particular thymine. Okay, come back to this. Density of DNA decreases on heating, and see the next choice, adenine plus thymine, cytosine plus guanine is not constant, both A and B. Okay, so in so many questions you can you can see the choices like this. You have to choose 2 or 3 choices at the particular time for the particular question, and see one by one. When here we are talking about the double helical structure or double helical nature of the particular DNA. Okay, so in a DNA you have to remember there are 4 bases present. I told you it is made up of adenine, guanine, thymine and cytosine in a particular composition for the particular sequence. Okay, and we all know adenine always pairs with thymine, guanine always pairs with cytosine. Adenine always pairs with thymine, guanine always pairs with cytosine. Over here we are mentioning this as hydrogen bonds, not as equal to. Okay, adenine pairs with thymine by means of 2 hydrogen bonds, guanine pairs with cytosine by means of 3 hydrogen bonds. Okay, so remember this and come back to this. Adenine equal to thymine, cytosine equal to thymine. This is not a correct thing. Cytosine is equal to guanine, not a thymine. So this is a wrong choice. Adenine is equal to thymine is correct, because as I told you, adenine always equal to, adenine is always pairs with thymine. So adenine and the number of adenine is always equal to thymine. As I told you, you have to remember this thing. This is a double helical nature, double stranded structure. Okay, so in a particular one strand, if the adenine is present, the opposite strand must possess the particular thymine, then only it will make a hydrogen bond over here. So while you are counting the number of adenine, it is always equal to number of thymine. Okay, so the number of adenine is always equal to thymine, number of guanine is always equal to cytosine. This is what you have to remember. So over here adenine is equal to thymine, up to this correct, but cytosine is not supposed to be always equal to particular thymine. Cytosine is always equal to guanine. So this is a wrong choice for the particular question. So over here, density of DNA decreases on heating. So what is density? So I told you it is a double helical structure and it is being filled with the nitrogenous bases connected by hydrogen bond. So what will happen while you are heating the particular DNA in a solution? That heating of DNA will denature the particular structure. Why it is denatures the particular DNA structures? It is going to break the hydrogen bonds. It is going to break the hydrogen bonds in between the particular nitrogenous bases. So if there are no hydrogen bonds, it will release the 2 strands apart. So it will increase the density of the DNA. So this is a wrong choice. Okay. So both A and B are wrong. So you can eliminate the particular choice. So answer is A plus T and C plus G is not constant. That is the correct answer. And we are going to explain the particular thing in the next question. A plus T and C plus G is not constant. Okay. Come back to the next question. I told you. Which of the following nucleotide sequence contains 4 pyrimidine bases? Okay. So this is a very straightforward question. So over here you have a DNA sequence like this. Okay, they have given some sequences. I told you we can write the DNA sequence by means of their alphabets. I told you there are 4 bases: adenine, guanine, thymine and cytosine. Suppose you want to write a DNA as a sequence, you have to start from a 5' end to 3' end. Okay, then you can write the sequence like this. You can write the sequence like this. Okay, so 5' is refers to a particular end, and from the 5' you are proceeding with the direction to the 3 prime, you have written the particular DNA in a sequence in a particular paper, a written form of the particular sequence. So over here there are 4 sequences given to you, there are 3 sequences and a choice given to you, and you have to choose a sequence which contains 4 pyrimidine bases. I told you there are 2 types of bases, purines and pyrimidines. What and all purines? Adenine and guanine. Thymine, cytosine and uracil are pyrimidines. Okay. So with this knowledge you have to count the pyrimidine bases in the particular sequence. So over here, so what are the pyrimidines? Thymine, cytosine and uracil. Thymine, cytosine, thymine, cytosine. Okay, over here cytosine, cytosine. Only 2 pyrimidines are present, so this is not a choice. So over here uracil, cytosine, uracil, only 3 pyrimidines are present, so this is not a choice. So we have eliminated B and C, so B and C of course. So over here you can count 1, 2, 3 and 4 pyrimidines. There are thymine, cytosine, so the correct choice over here A. Okay, this sequence contains 4 pyrimidines. Okay, see the next question. So here we are going to explain the previous question. A segment of DNA has 120 adenine and 120 cytosine bases. Okay, so the question is the total number of nucleotides in a particular sequence. So as I told you, so here you have to remember the term Chargaff rule. Chargaff rule. So according to Chargaff rule, as I told you, adenine, number of adenine is always equal to thymine. The number of guanine is always equal to cytosine. Okay, why this happens? As I told you, adenine always pairs with thymine, guanine always pairs with cytosine. So that the number of adenine is always equal. Okay, you have to remember the term equal. Always equal to thymine, guanine always equal to cytosine. Okay, so I told you in the previous question, the A plus T and C plus G is not constant. So why we are calling this as not constant? I told you. So from this statement you have to remember one thing very clearly. The number of adenine may not be equal to guanine. The number of thymine may not be equal to particular cytosine. Why? Because they are not gonna make hydrogen bond with each other. Okay, so the number of adenine may be differs from the number of guanine. The number of thymine may be differs from the number of cytosine. So that is what we have explained over here. The A plus T content and C plus G content is not constant for the particular species. Of course, we can identify the species based on a particular ratio. Okay, for each and every species, for example, for each and every species, the A plus T and G plus C, the ratio between these two, the ratio between the adenine, thymine and guanine, cytosine ratio is constant for a particular species. For each and every type of species, this ratio is constant. That means the number of adenine and the number of thymine is particular. The number of adenine and thymine is constant for the particular species. In the same species, the number of guanine and cytosine constant in the particular same species. Okay, so this is what explained over here. The ATCG is not constant. Okay, that is what you have to remember. That means the number of adenine is not equal to cytosine. The number of thymine is not equal to guanine. This is what you have to remember. Come back to the final question. As I told you, a segment of DNA has 120 adenine and 120 cytosine, and you have to find out the total number of nucleotides over here. So from this you can easily predict. So number of adenine over here is 120. So what may be the number of thymine? As I told you, the number of thymine is equal to adenine. Adenine always pairs with thymine. So the number of thymine is of course 120. Why? The 120 adenine base is gonna make bond with 120 thymine. So another hint over here, the number of cytosine is of course 120 over here. Cytosine always first with guanine. So the number of cytosine over here 120, so the guanine must be 120. And the question is the total number of nucleotides in the particular sequence. You are gonna add up all the things. The sum of this 120 plus 120 plus 120 plus 120, 480. Okay, so 120 is wrong answer, 240, 60, the answer is 480. Okay, so this is how you have to select the answer for the questions you can expect from the particular topic, the nucleic acids. So in the next classes we are going to see the question from the some other topics from the particular biomolecules.